Hello, everyone. Good to see all of you again after a long gap. We are back with yet another episode on anthropology. And specifically speaking, we will discuss biological anthropology in some detail. But keep in mind, our anthropology option is not just about biology per se, but it is about biological anthropology, where we emphasize more on the applications of these biological aspects to humans, to humanity, right? That's what anthropology is. It's a discipline of infinite curiosity about humans. So that's an important sort of a distinction to keep in mind. So a biology answer isn't necessarily a biological anthropology answer. And one more thing, most of the students probably think that biology portion is going to be taxing, it's going to be slightly challenging, but trust me, that is not the case at all. Biology is in fact quite easy, and in fact a lot of fun as well. And you yourself will realize this thing after watching these videos on the playlist. So in this playlist, we will start from the very basics and try to sort of help you build some interest and more importantly, help you build a strong foundation when it comes to biological anthropology. So without any further delay, let's get started.
Now I'm going to go into RH. In this one, what do we have? Similar to that, we have positives and negatives, right? So again, this was a little later, it was discovered after this. So this blood group is slightly more complex genetically, okay, because it actually involves a lot of different antigens on the surface of the red cells and all that. But leave that for now. Now there are two closely linked genes, and this is from your chromosome 1 and all this. Here, the inheritance pattern is very, very simple. So essentially, we have two alleles, okay, which is your capital D and lowercase d. So capital D is plus and this is negative, right? So if I say capital D, and capital D, now do this really quick and tell me the genotype and the phenotype. Okay, which is basically my positive, and negative. Sorry. Okay, so basically like this. Right? Did she get it right?
Yes. Yeah. Yes, sir.
Okay, you did. Okay, so something along these lines. If you have a particular blood cell, something like this, if it is A, then it will contain antigen for A. This will be your A. This will be your A, something like this. If it is B, what would it be? It will be B's, B, B, B. What if it is AB? A, B, A, B, A. You get the point, right? So it will be something along these lines. Okay, so you have antigen of A and you have antigen B as well. Now, not A, A comma B. Now, what will be the antibodies that you need here?
No, okay. Don't know, sir.
So what if I insert antibody A here into this blood group? It'll start fighting off all your A, right? All your A's. If I send anti-B here, you will start fighting off all your B's, but these are part of your own body and you do not want to be doing that. So what would you do? You would have no antibody A, no anti-A, no anti-B. Did you get it?
Yes, sir.
Now, if I have O, it's empty. You don't have any of these antigens. But let's say if I am O and you send in blood A or blood B. So this is Revati, right? If I send O blood group into you, what do you think will happen to you? Sorry, not O, A and B.
It should be anti-A and anti-B. Otherwise, it will...
You need both of these things so that you can fight off your invaders. Okay. And in terms of antigen, you have none of these. Okay. Is that clear?
Yes, sir.
Okay. So this is your antigen antibody. So essentially what is it? Antigen is something that is present on your, this. Can you see these? Right? This is something like that I've drawn here. However, you need a mechanism to fight off any foreign body that is not part of your blood. And that is all these things kick in when you talk about blood transfusions. It's like when you have to transfer blood from one body to another, right? So in that time, you need to keep these things in mind. Are there any antibodies that are present? Okay, that will probably start the rejection of the blood group that you're sending in. These kinds of things will come in handy. But just think of it. If it is O, you don't have any antigen, okay? And anything that you send in, A, B, whatever it is, sorry, A, B, these things will need to be fighting it off, right? So you will have anti-A and anti-B. If it's a blood group B, if you just remember your own blood group, I think that will suffice, because you can factor the rest later. Okay, you can derive it. You get the point, right?
Yes, sir.
Okay, so no questions on this? See, generally,
No, sir.
individuals with O blood group, right? They don't produce antigens ABO. So therefore, their blood normally will not be rejected when it is given to others with ABO blood types. You understand? And that is why it is called as a universal donor. Type O blood are universal donors. Have you guys heard of this thing? Especially when you're doing this blood donation or transfusion.
Yes, sir.
Yeah.
Okay. So, and essentially the whole idea here is that, you can even just, if you want to derive it, you can just, and I think this is something that we've already done. You can make, possible alleles, allelic combinations. What is allelic combination? And then try to see what are the combinations that could happen. Okay. And what would be the phenotype? All of these things would come in handy. You'll understand it better. Now, similarly, I don't think I have any more space. I'll go to the next one. So if I'm talking about RH, okay, so what is RH blood group? So just some funda, 1940, Carl, Land, Steiner, okay, Alexander, okay, so discovered by these guys, okay, and it's been, what, around 40 years since the previous one. And this is the one that gives you what you normally hear as positives and the negatives. So both of you are positive. Have you ever heard of anybody with negative blood group amongst your family, friends?
No, sir.
Okay,
Can't
it is a
remember.
little, okay, it is a little less common in India. Okay, but I know friends, like for instance, my close friends, a couple of them have negative blood group. Although one of the girls, I think her ancestry is to Uzbekistan, but I'm not sure how far back it is, right? They are like Indianized Muslims, but they're from the western part. Now, generally what you'll see is that, genetically it's not very easy to understand this. It is actually a little complex. I'm not going to write complex here. However, the inheritance pattern for just one particular antigen, which is your D antigen that we are going to talk about, that is pretty simple. And for us, that is the only thing that really matters. That is the most important one as well. Okay, and basically it's like there are two linked genes. And I'm sure you guys remember what's a gene, right? And in chromosome number, these are not something that you need to know. Okay, but just some extra funda. Now the point is, if I tell you, because this has been asked, okay, RH factor, there was a question number, was the first question in 2016, I believe. Okay, and it's a 10 marker, right? So let us say we've already drawn this, right? So you've understood. So I'm not going to talk about the Punnett square. You know what is law of recessive and dominant. Okay, capital D. Okay, and this is lowercase d, right? So which is dominant? The one that is portrayed with a capital D is going to be the dominant one. Okay, now now that I've given you the whole thing, basically you will have similar kind of things, right? Now if I ask you a question, why is this important to know? And I'll try to ease you guys in, because I think it has been a while since you probably took a look at bio, right? And I'm not sure how comfortable you are. In case you find it a little, if I'm going a little, I'm hoping that I will not be delving in too deep today, but in case you feel like that, just do let me know, okay? And then we'll take a step back and do a little more surface level work, and then come back and slowly delve in subsequent lectures. Have you guys heard of this thing called RH incompatibility?
I think it has something to do with RH negative, sir.
Yeah, so you heard something about it. So what is it? It's also known as hemolytic
Yeah.
disease of the newborn.
So the mother has RH negative and the baby is RH positive, then that's where the issue comes up.
Perfect. Now see, you've answered this question like a layperson, right? The most important thing is to know what is the lay perspective, right? So once you understand what it is, then you can actually try to explain it using all the bio that you know, right? Now you are, armed with certain important concepts in bio, right? So you know what is a cell, what is a chromosome, what is a DNA sequence, what are the genes, all of these homozygous, heterozygous, genotype, phenotype, all of these things you know. Now your ability, and plus RH blood group system, ABO blood group system, Mendel and his laws. One second, try to let somebody in. One second. Hey, just call me or message me, okay? I've already logged you in now. Okay, so just now, yeah. If you can see, just let me know, okay? You can. They can, but they do participate as well. So you can go ahead and mute on top, okay? Just, you can't hear me yet. If you can hear me, let me know. Okay, so where were we? I forgot, like, my train of thought.
RH incompatibility. Yeah, RH incompatibility. Just trying to understand why it's incompatible.
Yeah. So essentially using all the concepts that we already know, we want to be able to explain it in a manner that you can put it in a couple of pages, right? So what are the keywords here? So you have your antigens and antibodies, and you want to explain these things, right? Now, this particular one that we're talking about, we are going to talk about factor D. And one of you mentioned that RH incompatibility is when you have a case of positives and negatives. So there is some incompatibility. What is this incompatibility? See, so factor D is, we're talking about D antigen. D antigen is what we're going to be discussing. So capital D is what? Positive. And lowercase d is going to be your negative in terms of your phenotype, right? Now just think of it like this. ABO, we just talked about it in a very, let's say for the sake of it, it is sort of comprehensive, right? You understand ABO groupings. You know that there are naturally occurring antibodies depending upon what is your blood type. Okay, and where does it occur? It occurs in your plasma. They're also known as immunoglobulins. It doesn't matter, these keywords, and you don't have to throw those things in. The idea is to ensure that you can explain complex topics and concepts in a very simple manner. So in ABO blood group, so you have these naturally occurring antibodies, where in your plasma. In RH blood system, you don't necessarily have those. There are no naturally occurring antibodies to this RH factor in your plasma. However, and it looks something like this, like how I've drawn this here. Take a look at this. I'm going to draw something along these lines. So let's say I'll draw it a little better. Okay. So let's say this is your, what is that? Somebody tell me. Anyone? What do you see on the surface? Can you guys still hear me?
Yes.
Yes, sir.
So what is it? what is it that I've drawn?
And is it antigen?
Yeah, so we are talking about your D antigen, and this is present on your red blood cells, right? So basically, there are certain kind of proteins that are present on top of your Rh positive. However, what I mean by negative is the absence of those. Okay, so if I do this, essentially it is without your antigens. This is your Rh negative. Now, this is going to be independent and they're going to work just fine. However, when there is a blood transfusion or when there is exchange of blood through the fetus into the maternal body, the maternal bloodstream, that is where this entire Rh incompatibility issue can throw in the problems. So let's take a look at blood transfusion. When you have Rh negative and if you are exposed to Rh positive blood, what would happen? This exposure will result in production of antibodies. So let's say blood transfusion. If you have blood transfusion, what will happen? It will result in exposure okay. Whereas in APO, what did we see? There are some naturally occurring antibodies that is present in your bloodstream, in your plasma, right? And this, essentially, these antibodies look almost like a Y shape. IgG, immunoglobulins, right? And they'll have some sort of receptors, okay, to bind where they have to bind. And then fight off those invaders, something like this. So this is like your IgG, immunoglobulin G, antibodies. But this again is not naturally occurring. It occurs only for Rh negative blood, which has been exposed to Rh positive blood. And this is something that I want you to understand. Did you get it? If you're a naturally Rh negative person, for instance, you mentioned that one of you is a B positive, one of you is O positive. Vinod, what is your blood type?
O positive.
Okay, so it's all positive here. So forget that. Let's say there is somebody with Rh negative. Just 1 second. In class, I'll call you back. Exactly. So if you have somebody who is Rh negative, that person does not have a naturally occurring antibody in their blood plasma. But however, if exposed to Rh positive blood, either through blood transfusions or through like fetus, right? Fetal blood being exchanged, in those cases, there will be the antibodies being produced by the body, right? So that is clear so far? Is that clear? So I have said nothing more than what Revati had already explained. Right? I've said nothing more than what Revati has already explained. The only thing that I've done is that I've drawn some RPCs and I've talked about antibodies. Antigen, antibody. That's the first thing that you want to bring in, right? Because we are talking in terms of. Can you guys still hear me?
Now, yes.
Okay. Actually, it's some big rains going on here, heavy. So I might have some issue. But hopefully, in case you can't hear me, just let me know. Okay. So whenever there is Rh positive father and there is an Rh negative mother, there is a potential complication. So what is the combination? It is not the other way. It is in one way. And I'll explain this again. So whenever there is Rh positive is father or mother? And I say potential. It does not mean if it is a short complication, then obviously possibly, there'll not be too many marriages between Rh positive and Rh negative. So, and this is that complication, your hemolytic disease, right, of the newborn. Now that you've understood so far, let's just go and dive a little deeper. So if I say, for instance, if in case I can draw a woman, and I hope you can make some sense of this. This is essentially a pregnant woman. Is this clear? Let's say I'm drawing a baby, right? So if I have a combination of this sort, so let's say this baby, because we already saw Rh positive is dominant. So if the father is Rh positive, even a combination that is capital D and a lowercase d from the mother and a capital D from the father is still going to be an Rh positive child. Okay, so this Rh positive here, child, okay, and Rh negative mother. And this is Rh positive. Why? Because courtesy of the dominant allele sent by the father, right? Now the risk is, let's say at some point during the pregnancy, there is feto-maternal hemorrhage or blood leaking from the mother's fetus into the, from the fetus into the mother's maternal blood circulation. Now this doesn't always happen, and definitely doesn't happen during the first part of the pregnancy unless there are some issues. However, this could happen towards the end of the pregnancy, okay, and definitely during pregnancy, definitely during delivery. So during delivery, it is guaranteed that some of this fetal blood from this particular child via the torn placenta is going to get into the mother. Is that clear? Do you want me to repeat this again?
Yeah, clear.
Yes, sir.
Clear, right? So what I'm saying is feto-maternal hemorrhage, blood leaking from this particular fetus, which is Rh positive, into the mother, which is Rh negative. And this may not happen in the starting part of the pregnancy, the initial part of the pregnancy, and it may happen towards the end of the pregnancy, but definitely it's guaranteed when you have to push out the baby and, when the baby is born. So at that particular time, there will be some fetal blood through the torn placenta that is going to enter into the mother. So if you guys get disconnected, join back in, okay? Because I think it shows less than a minute. So let's say I'm going to draw the mother again. So what has happened? Now, Rh positive is entering, and it is entering, right? And you can even say that it is during pregnancy. Some of this Rh positive blood cells will enter into the mother's, blood circulation through the fetal maternal hemorrhage at some point, even if it is towards the end of the pregnancy during delivery, though it is definitely guaranteed. So the body has to come up with some sort of a response, right? What will be the response considering this body is going to be an Rh negative mother, right? Just tell me you have certain antibodies, and we just said here that at one point, if it enters into the bloodstream of Rh negative, right? If they have some exposure, what did we talk about? What exactly would happen?
Production of antibodies, sir.
Exactly. So I'll just go to the next slide. So essentially, what you're seeing here is that you have certain production of antibodies that is guaranteed. So when you have that exposure, you're going to have some, and this is the Y that we talked about, right? And here you'll have some receptors, right? So essentially the mother is now getting sensitized, meaning the baby is born, but mother, okay, after the baby is born, the mother is going to develop. Once this thing has happened, you will notice that the body's response is the production of IgGs or antibodies against this Rh positive blood that it has been recently exposed to. Okay, but it's okay. It's not a problem because the child has already been born. This why, okay, these antibodies are now going to fight this Rh positive blood group of the child, right? However, the mother's bloodstream now contains something that it did not contain previously. Okay, so what I'm trying to say is the first child has been okay. No problem at all. However, the next time, let us say, with the same sexual partner, right? If it is going to be another capital D that is being sent in as RH, in the RH blood group system, what would be the change here? So let us talk about the second child. So again, the same mom. And there's another child. And what is this blood group? It's again an RH positive. And this is negative. What do you see different? See, this migration of antibodies is not something that we want to prevent, right? You do not want to prevent that because it is required. It's a natural process. In fact, it is essential in some cases, right? Especially, in terms of like antibodies against bacteria, viral disease, all of these things, you need to have this transplacental migration of these antibodies. However, now we are left with a case in which the mother also has antibodies present here against this Y, right? Against this RH positive blood group. So let us say, if these are going to be my antibodies, what is their duty? Is to go and fight anything that is positive, RH positive, right? If it is positive. And this is something that we want to avoid. And this is what they call as destruction of your red blood cells, or it hemolyzes the fetal red cells, red blood cells. So the baby will develop, thanks to the mother's antibodies, Do you understand this? It did not happen with the first child, but it happened in the second child. I hope this is not too fast for you, because I know that today is your first day after very, very long in bio. So I'm trying to go slow and I'm trying to not complicate it any further. But so far, have you understood it? How and why the problem is only with the second child, not with the first child?
Yes, sir, I understood that. But could you just repeat how the baby will develop hemolysis? Or
How the
could you just
baby
repeat
will
that,
develop?
sir?
Okay. Any other questions?
But even in during the first child, there will be certain antibodies that will be generated, right?
Right. Certain antibodies. Certain antibodies. Okay, good. Any other questions? Nothing, right? See, okay, so the first question is about how will the child develop hemolysis? Okay, now for that you have to understand what is hemolysis. Okay, that is generally outside our pay grade, right? Or basically it's outside the scope, because we don't want to dwell so much on the medical aspects of it, but you want to know a little bit enough. Okay, and certain antibodies, of course, as I said, and this is a fact, that transplacental migration of antibodies is not only natural, right? It is essential in some cases, right? Especially to ward off any kind of diseases. And the mother's duty is to produce that and then send it to the child. However, when you have RH negative of mother and RH positive in the particular child, okay, and you have something that is there to destroy RH positive, you do not want that in the blood, because it is going to go and try to, destroy the red blood cells of the child. It is going to try to destroy the red blood cells of the child. So this antibody is fighting against what? It fights the baby, and this is something that you want to avoid, because baby as in the baby's RH positive blood group, right? And that is the whole point that we saw in the first one also, right? If there is a transfusion of similar blood group, okay, there will be no problem, because the antibody is now going to reject that. However, if you send something else, okay, if a B positive person is going to have blood being sent into, or transfusion of that, then obviously the blood has to find a way to fight it and reject it. Okay, so here is a situation where you do not want those antibodies trying to kill something. This is not a foreign object, right? This is a child that you want to deliver. Okay, is that clear? And in terms of
Yes, sir.
hemolysis, right? So see, hemolysis is there's a field called hematology. Okay, somebody who studies blood really well. Actually, one of my aunt is, anyway, the point is she had given me some insight into this. Essentially what happens is that the red blood cells of that particular child, right? What is the job of red blood cells? In red blood cells, 95% of it is going to be your hemoglobin, and there's something that we've already seen before, hemoglobin, right? And the duty is to ensure that there is oxygen carried by the blood. Okay, it's an oxygen transport system. This will ensure, because of the destruction of your red blood cells, there will be reduced carrying capacity by the blood of this oxygen, right? So reduced oxygen carrying capacity, right? That becomes much difficult for the child to, so because of this reduced oxygen delivery to the required body parts, like your brain requires a lot of blood, oxygen, there are other parts as well. So in that baby's body, the heart in that baby's body will have to beat harder to try to compensate for that, because it is not getting enough oxygen. Why it is not getting enough oxygen? So to make up for that, this has to compensate and it will beat harder. So this can lead to fetal cardiac arrest, okay, or cardiac failure. It has to pump with such great blood pressure, right? And probably like much faster, so that oxygen is delivered. So what the red blood cells are supposed to be doing, if you ensure that the red blood cells are not strong enough to carry the blood, to carry oxygen, or they're not just working right, okay, so they will have to beat much harder, pump much harder, and this can lead to fetal cardiac failure. So basically this is a case of anemia, right? And we also know that during this hemoglobin, when it is being destroyed, one of the byproducts of breaking down of hemoglobin is something called bilirubin. Do you want me? Okay, I'll write these for you. So what did I mention? So because you wanted it. So can I just say anemic? Okay, and red cells. Will you remember this, and will you be able to write? Or do you want me to make down a few points? Reduced O2 delivery. I'll say reduced oxygen carrying capacity. And what will be the baby's response? Cardiac failure, right? Because of this entire red cells struggling with these antibodies working over time. And when this hemoglobin is, so let's just put this as your first, right? So how does the baby develop hemolysis, and what exactly does it mean? Fetal cardiac failure in that second child is a big possibility, unless you try to find ways to ensure that these antibodies do not destroy these Rh positive, blood group, right? Now, another one is basically your hemoglobin. You know what is your hemoglobin, right? What does hemoglobin do?
It carries oxygen to all cells.
Right, and this is present in your red blood cells, right? So when it is broken down, one of the byproducts is bilirubin. Have you guys heard of this? Okay, so bilirubin is one of those compounds that is produced as a hemoglobin breakdown byproduct. And during this particular process, as the number of antibodies try breaking down this red blood cells and the hemoglobin in that child, there'll be lots and lots of bilirubin being released. And bilirubin levels is something that you would have heard in case somebody has jaundice or something like that, right? So, so that is that bilirubin, right? So this is also toxic to the brain. It can lead to brain failure. Bilirubin, toxic to brain. Jaundice. there are certain communities that have more of a presence of negative blood group, and in them the chances of having this issue is a lot more pronounced, right? Again, how will it develop hemolysis? See, red blood cells are basically produced in your liver, your spleen. This results in your liver having to work overtime, right, to produce enough RBCs to make up for the inability of the present red blood cells to send in enough oxygen. So this is something called, so your liver size will increase, its function, it's basically its capacity will decrease. Okay, so this will also result in something called hematomegaly, right? So this is another one. So liver getting affected. Okay, because why? Big role in producing RBCs. Generally, the most severe cases will see intrauterine deaths. I hope my handwriting is a little legible. See, the whole point is, why has this entire thing occurred? It is occurring because in the first pregnancy, right, you did not have any of these complications because, let us say, on the day of the delivery, you have these antibodies migrating because there is, okay, a little bit of exchange of fetal blood and the maternal blood. Once there is exposure of the maternal, I think in the starting of RH, when you were discussing it, I mentioned, right, you do not have naturally occurring antibodies. However, an RH negative antigen, where is this? If I have RH negative, does not have any of these antibodies. However, when you are exposed to RH positive blood, it will result in production of antibodies. Now tell me, once the child is just getting born, there is some migration of antibodies. Why so? Because there is exchange of this blood. So there is exposure of RH positive blood into the mother. So production of antibodies has now occurred. Now, let's say after 3 years, they have another child. And this particular child has to face another big challenge, which is the presence of millions of these antibodies that is already present. However, if this was not there, then the child could be normal, just like the first pregnancy. Because until the end, until the day of the delivery, sometimes it could be even prior, but generally, let's say, if it is on the day of the delivery, what would happen? Only then there is an exchange of blood. Only then there will be an exposure of RH positive to this RH negative mother. Only then will it start producing those antibodies. If the child is already born before the production of these antibodies are not working overtime to destroy these red blood cells in the child, or resulting in all of these kinds of problems. Is that clear? It is because of the introduction of these or production of these antibodies in the mother's blood is why you have, in more severe cases, intrauterine deaths. Is that clear? Just the presence of these antibodies that are being produced in the mother's body has resulted in so many of these complications. Is that clear to all of you?
Yeah.
So can we prevent it by saying that Rh factor antigens or this D antigen, okay, are not going to get into the maternal circulation? Can we do that? Is that a possibility? That is not a possibility. There will be some exchange, right? It will be some exchange regardless of the way of your delivery and whatever it is. So generally what they do, this is not something that can be treated this particular way, right? Or at least it can't be cured. But generally what they do is that you want to ensure that these antibodies that are there already, right, if they don't act up like this, right, the body produces these antibodies, right, the body will produce these antibodies when it does not have these antibodies and there is exposure to Rh positive. So what they traditionally tend to do is that around 28 weeks of pregnancy, they give AntiG injection. So basically they're introducing artificially some immunoglobulins. Now, when you're sent in through injection, right, some sort of these antibodies, you already have these antibodies. Your body does not find the need to produce antibodies separately. But these antibodies work in such a way that they do not last long. Definitely not until the next pregnancy. Do you guys understand what I just said?
Yes, sir.
What about the other two?
Can you repeat the last part?
See, they're trying to give you some sort of anti-D injection, right? So now if there are lots of these antibodies already there that we are putting in artificially, then as soon as the fetal cells, right, from the baby, they would be mopped up or taken away by these antibodies that we have injected into the mother's body. So that is why around 28 weeks or something like that, right, they put in this anti-D injection so that the mother does not need to produce these antibodies by herself. That will last much longer until the next pregnancy.
Oh, okay.
Okay. And they also give an injection of this anti-D antibodies, right? It's like a form of passive immunization of the mother already. And this is done even immediately after the birth, okay, within the first 24 hours, when there is a new exchange of blood that is already happening. So later part of the pregnancy, when I mentioned that there could be some exchange, and definitely during the delivery period, when there is definitely going to be an exchange of this blood, you want to give some form of passive immunization to the mother, introduce these, pies that we have drawn, so that the mother does not have to produce it, right? The mother does not have to produce it, but it is passively put in by us. So any of these positives that have come out will be taken away or cleaned up or mopped up by these Ys. So the mother does not have any of those positive anymore. And also these Ys or these immunoglobulin G or these antibodies are not going to last much longer in the blood either.
Sir, but these antibodies don't affect the baby.
Okay, tell me why.
No, I'm asking you, sir. Will they not affect the baby? Because they don't last longer.
See, so what I'm mentioning is we are in this particular phase, right? So this is the right after the birth, after the delivery, when there is exchange of Rh positive and Rh negative between the mother and the child, right? So the body will produce these antibodies, right? However, at this particular point somewhere here, right, before the introduction of these Rh positive into the mother's, maternal circulation, what you want to do is that you introduce those passive immunization, you introduce some antibodies that will not last as long, okay, but they'll still do the job to clean up any of these Rh positive that enters into our blood. Got it? So you need antibodies to clean up any of those Rh positive because incompatible with her blood group. So it'll do that particular job, but it'll not last as long, which means any of the fetal red cells, which is Rh positive, that enter the mother will be quickly, agglutinated or hemolyzed or just destroyed, okay, by the antibodies through this particular injection. So this is a form of passive immunization that they do. So within a few months, the mother would have gotten rid of all of these antibodies that have been introduced through this, passive immunization, this injection. However, if she had, developed this thing by herself, it will be much longer lasting. And that will be a problem for the next child, as you can see here. So by the time the next child is born, these antibodies would not be in the mother's blood anymore. So it will be the same case of this, right? It will not get affected until the delivery process, when there is a final exchange of blood between the mother, which is Rh negative, and the child, which is Rh positive. So what we have done is ensured that she will not have, or she wouldn't have produced her own antibodies. So by giving this anti-D, okay, or anti-rhesus factor D, these immunoglobulins, right, at whichever period where doctors see that there will be an exchange. Generally, it's given at 7 months and within the first 24 hours. This will mop up, almost like a sponge, right? It will mop up all the Rh positive fetal red blood cells that will enter the mother's body. Okay, so that way the mother is safe. No Rh positive entering the body is going to be a problem. Okay, by giving passive immunization or like outside immunization and not allowing her to produce her own. Is that clear?
Understood, sir. Yes, sir.
Yep.
Vinod, you've got it as well? You've got it?
Not that it's relevant. I have a doubt.
Mm-hmm.
The concept that you just explained, I got it. See, but this is in the first case, in the first baby case, due to the placental breakage, the blood transfusion occurs from baby to the mother. Correct?
Repeat that again.
So that's why during birth of first child, if the mother's and baby's blood group doesn't match, I mean to say that positive and negative,
Yes.
then due to the placental breakage or something, the blood transfusion from baby to the mother occurs.
Yes.
During delivery. So that's the only time, right?
Yeah, generally in the first part of the pregnancy it doesn't happen. See, we don't have to think about like when exactly it's going to happen. It really depends on the complication, but generally you cannot avoid it during delivery. It can happen towards the
correct. So
fag
you
end
can't
of the
avoid
pregnancy.
during delivery.
Yes.
Correct. You can't avoid it during delivery. My thing is to avoid that. Again, not that it's relevant. What about cesarean cases? They can opt for cesarean, no? So that there is no breakage and all.
See, the exchange of blood is something that you cannot control. Okay. That entire that fetal thing, that wall is there, right? With your amniotic fluid, your placenta, your the chorions and all that. The baby will be cut out and taken from it. So during that, in pregnancy or delivery, you will not be able to stop this exchange.
Okay. Fine.
Okay.
Okay. Fine.
So, but generally they give it, at 28 weeks itself, in case anything happens. Okay, there is some sort of a rupture or whatever it is. If you want to avoid this, right? If this is going to happen, the mother's body is going to find a way to be able to fight off those red blood cells, right? By production of her own antibodies. The only thing is that we want to try to make her not produce it, but we'll introduce it in such a way that it is not those long-lasting ones. Okay, but at the same time, it can fight off those Rh positives so that it is not a problem for her blood. Right?
Okay.
And when it comes to this particular situation, these antibodies will generally after a few months, okay, not be there anymore. So it will not be a case of this, it will again be a case of this again. And only during the delivery of the child, when there is a rupture, again we'll go and explain the same thing. Okay, at that particular point, if they've given those, passive immunization again, then she will not have to produce her own antibodies for it. The whole point is, again, bringing it back to anthropology, is that, see, what have we done? We've understood this. And this is a very good example of explaining your law of recessive and dominant and recessive, which is, given by Mendel, right? So capital D, lowercase d, and, all kinds of combination that can happen. Now, one of the reasons why, if I ask you, why does it really matter? We as humanity, why are we so interested in it? We're interested in it because it is sort of important. There are certain blood groups that are present. And if you look at the distribution, in India, a little less, okay, but generally in this North European population, the Western European population, basically the white Caucasian population, Rh negative is a lot more common. So this kind of a disorder or disease, hemolysis, is seen a lot more. So by understanding this thing, there are certain benefits. So just by, and you may have not known about Mendel or, the capital D's and the lowercase d's and the recessives and dominants, but just by knowing so much, okay, you've applied it to your blood group and understanding what could be the possible problems that you could face and what are the solutions that you can provide just by understanding some basics of this. Essentially here, you're saying that the time the mother is exposed to the Rh positive, okay, the red blood cells and all that, is very short. She wouldn't have time to produce her own antibodies. And she does not have to also. She won't produce her antibodies against her second child. Okay, so this particular Rh incompatibility or hemolytic disease of the newborn, right? It's also called erythroblastosis fetalis. Okay, so there is no case of this as well. So by doing this and avoiding this thing, you have basically avoided this erythroblastosis fetalis, even if the mother and the father belong to Rh positive father and Rh negative mother. So this is again a perfect example of where prevention is better than cure. Once the child is born or is about to be born, you cannot control these. Right? If there is less oxygen carrying capacity, the heart has to do nothing but try to compensate and try to provide oxygen. It is going to beat harder and louder and faster and whatever it is. Okay, there's going to be immense stress that is put on that little heart. Okay, if you're breaking down hemoglobin, it is going to produce bilirubin. There is nothing you can do about it. So this is a perfect example of prevention being better than cure. So that is the possibility. One of the benefits of knowing all of these things is that there is possibility of genetic counseling. Okay, a person and his wife Okay, or two sexual partners going and meeting a genetic counselor. The first thing the easiest thing they can showcase is that, okay, there is Rh negative mother and Rh positive father. This could be a possible problem, but this can be resolved with so and so and so. So there are so many benefits. In fact, it's potentially saving the lives of how many children? So many second children and so many fetuses.
And so you're using possible complications, right? When possible. The father is Rh positive, the mother is Rh negative.
Yes. Now, why did I say it is possible? Yes, go ahead.
So that means that Rh positive, Rh positive is not a dominant allele. Always.
No, Rh positive is dominant. However,
always.
Rh positive is positive. Is dominant. So capital D is dominant and lowercase d is recessive. But still, the child can be Rh negative. Can you tell me how?
No, that's because of...
What could be the combination that could result in the child being still Rh negative and not the Rh positive that we've seen here? It's open to all
25%.
of you.
25%. Wait.
Revati, Harika, is that true?
Yeah, 20%.
Yes, sir. Because if the father has uppercase D and lowercase d, and mother has lowercase d and lowercase d, then there is a possibility.
Exactly. Revati, is that right?
Yes.
But it is not always 25%, but it really depends on what the father has. The father could be heterozygous, but he could also be homozygous. If he's homozygous, then there is no way to prevent this combination. Yes, it can be capital D and a lowercase d.
Yes, sir.
So when you say 25%, that really depends on what combination of parents that you're talking about. Now, if the mother is to be Rh negative, she can have no other option but negative and negative. But the father has two possible combinations, the homozygous and the heterozygous. So when you say 25%, that is only 50% correct. That is based on your assumption that the father is capital D and lowercase d. Unless it is mentioned, you cannot mention it as 25%. Is that clear?
Yes, sir.
Vinod?
Yeah, correct. So even if mother is capital D and small d, then also the case varies, right?
If the mother is capital D and lowercase d, what will be the phenotype for it? You've just mentioned the genotype. Now tell me the phenotype for it. What is recessive? What is recessive?
No, see, phenotype, see, the capital D will be the visible characteristic feature.
Yes, but the mother is Rh negative.
Oh, yeah, correct. If mother is Rh negative, then she has to have DD, small d and small d.
There are only two combinations that can be. Father being capital D, and mother being lowercase d, or father being heterozygous, capital D, lowercase d, and mother still being lowercase d. Okay.
I got it. At one point, I got confused between the baby's genotype and mother's genotype. Actually, we are talking about mother's, when you're talking about mother's genotype, and she has expressed characteristics of Rh negative, then she, in her genotype, it is small d and small d. Correct.
Got it, right? Yeah. Now, so whatever it is, if you go step by step, okay, by starting with what are the combinations, what are the allelic combinations, what is the genotype, what could be the possible children, and then you take your Punnett square and then make those combinations, and then you mentioning ratio is very good, okay, because it helps you show that you're applying the concept rather than just ratifying some answer and then reproducing it in an exam. You're showing that in exam hall you're trying to apply that particular concept. It adds good value, okay, in terms of expression and presentation. Now, if you go logically, it'll be very easy for them to understand, even if you don't go into all the details that I've gone through. Okay, but just give an in summary about the possible benefits and bring in genetic counseling and talk about the lives, saving the lives of so many of these fetuses, especially the second and the subsequent births. Okay, and because it's so important, right? There are populations in which Rh negative mothers will be there. Okay, and it is not so uncommon like you think. Okay, and of course, all those children and families, they'll be really grateful for anybody helping them in being able to resolve some kind of a problem like this. So absolutely a perfect example. You can use this other places as well, of where prevention is better than cure. Anti-D, passive immunization, right? Any questions so far? So does this summarize this entire thing that we've been seeing, in terms of like what are the different blood group systems, Mendel's third law, the Punnett squares and these combinations, and again, applying it in a way that you understand, what happens when there is sexual reproduction, sexual congress, what exactly happens, what could be the problems, what would be the solution. So this, all of these things, once you understand, so your ability, so I'm not sure how many people would really understand it at this level. Okay, frankly, the answer is not many. Okay, but your idea is to ensure that there is some novelty to your answer. There is some uniqueness to your answer, that you write something that is different from other people's answer sheets. It is very easy to get bored in the same answers again and again, because at the end of the day, everybody is going to study for these 3 months and they will know something about every topic. Okay, 95% of the topics you'll be able to write something. But the problem is you will not be able to find ways to express it differently. But this will help you. So try to incorporate these things in your paper. And now it's your ability to utilize this when, where, how, in how much length. All of these things are up to you. Okay, so your ability to be very agile and nimble with this, as long as you're armed with this particular concept, with full understanding, it's going to do wonders for your score. Is that clear?
Yes.
Hope you guys are finding a biological anthropology playlist useful for your preparation. Hope this video helped reinforce your basics and also helped you connect a little better with the applications. If there's any other aspect of anthropology that you would like to know more about, do let us know in the comment section and we will try to make videos on those as well. Thank you for watching and we will see you in the next video.